Okay, let's finish up the photosynthesis chapter. And it's going to be a real short one. I didn't realize how far along the chapter I was. I probably could have done this all in one lecture. So we're just doing the Calvin cycle and then talk a bit about biofuels, I think. So the Calvin cycle, making sugar from carbon dioxide, functions like a sugar factory within a chloroplast. And it's in the stroma of the chloroplast. Remember, the light reactions are in the thylakoid membranes of the thylakoid sacs. The Calvin cycle is in between those stacks, what's called the stroma. So it constructs an energy-rich sugar molecule called glyceraldehyde 3-phosphate, or G3P, using carbon from CO2, energy from ATP, and high energy electrons from NADPH. So where's the CO2 come from? Comes in from the atmosphere through the stomata. Where's the ATP and the NADPH come from? This is something you could be asked about on the exam. Light reactions. It's a major function of the light reactions, or is the function of the light reactions, to make a lot of ATP and a lot of NADPH carrying high energy electrons. And they're going to go over the Calvin cycle. The plant cell can then use G3P as the raw material to make glucose and other organic compounds. So we're making G3P is what we're doing. The CO2 is being used with the ATP and the NADPH to make G3P. Then two G3Ps will come together to form one glucose. Because remember, each G3P is a 3-carbon molecule and glucose is a 6-carbon molecule. And then you can use these G3P to make other organic molecules as well. Plant oils, for example. Okay, here's the Calvin cycle. So CO2 from the air, right? So remember, with these processes, you need to know the input and the output molecules. So input to the Calvin cycle, CO2. To make a 3-carbon molecule, and here's all this ATP is used. So ATP is an input molecule, and that's converted to ADP and phosphate. So those are output. Then the NADPH comes in, supplies high energy electrons, and leaves as NADP positive. And so these G3Ps are fired up, and they leave. So some of them leave here, some of them go on. And when they go on, leave here, 2 of them put together to make glucose and other organic compounds. Some of the G3Ps stay in a cycle, get fired up with more ATP and make something called RUBP sugar. And that combines with another carbon dioxide to make the 3-carbon G3P again. And we go around and around and around. As long as we got ATP present and an ADPH present, we're good. And this will continue all day. And because we always got CO2 coming in from the atmosphere. But as soon as the sun goes down and nightfall, this will go on for a short time until the ATP and the NADPH get used up. And it comes to a grinding halt. And then, what does the plant have to do? Has to go to cellular respiration to make its ATP with cellular respiration. And now using oxygen instead of making it, and eating the food, breaking down the food that you made, the oils and the glucose, to make ATP to keep you alive until the morning when the sun comes up again. And then you fire up light reactions and you fire up the Calvin cycle. So sometimes the Calvin cycle is called the dark reactions because it's not strictly dependent upon light, but it is dependent upon light indirectly because once the sun goes down, the supply of ATP and NADPH peters out, and then you got to go to cellular respiration. You go to mitochondria to make the ATP to keep yourself alive. And same thing when in wintertime. Wintertime, these trees are making ATP with cellular respiration. They're not doing photosynthesis. Okay, So because even though it's light, the leaves are not. That's where the photosynthesis takes place. It doesn't take place in these deciduous trees. It doesn't take place in the trunks or the twigs. It takes place in the leaves. Okay, production of biofuels is highly inefficient. Working as biomedical engineers working to solve this by turning to the obvious example of evolution by natural selection. So you use a process called directed evolution, in which scientists in the laboratory determine which organisms are fittest. And using making biofuels involve algae rather than plants, because algae are much easier to manipulate and maintain within a laboratory rather than big plants, which you're not going to use trees. So a lot of biofuels are algae farms, is what they are. So microscopic biofuel factories. And this is, of course, completely renewable energy using algae. So in typical directed evolution experiment, which starts with a large collection of algae, they're exposed to mutation-producing chemicals. So we're going to get into mutations later on. These are errors in copying DNA. And there's always this background level of error, because the enzyme that copies DNA, called DNA polymerase, it's not perfect. There isn't any enzyme that's perfect. There's no process that's perfect going on in your body. So it makes errors all the time regardless of other factors. But there's some chemicals and agents in the environment that are mutagenic. They can speed up the rate of mutations, make these enzymes make more errors. And these are some mutagenic chemicals, and also lots of radiation can do the same thing. So the algae are screened for the ability to produce the most useful biofuels. Now, a lot of these mutations aren't going to do anything. Well, like I said, there's three results of a mutation. One, nothing. A mutation happens, an error is made, and nothing happens. And we'll see why that is when we get into genetics, and DNA structure, and what that really means to make an error. The second most probable thing that will happen is you'll screw things up, that they won't be able to photosynthesize anymore, photosynthesize it nearly as well, because photosynthesis is a complicated procedure, and you throw a random change into a complicated procedure, and you're likely to screw things up. But every once in a while, and you have to do this over and over and over again, because the rarest form of mutation is a mutation that actually improves the process. And that's what they're looking for. And so they can test all these algae, and these algae are making more glucose and more oil faster than the other strains. Maybe they got that would be a beneficial mutation. And if they're in a wild, they'll make more food than the next guy and be stronger and make more babies and pass this on. So they become more efficient in making biofuels by this directed evolution. So they screen for the algae that are doing better in this process than others, after subjecting them to mutagenic chemicals. And that's it, folks. So like I said, that was real short. That was so we're all done. So that would be 106 photo 2, and we're all done with acceleration and photosynthesis. And these two chapters will be on exam 3. That might be probably not until next week. We're doing fermentation again in the lab this week, and the next week we're doing chromatography, which is a more photosynthetic lab. Okay, so let me end this meeting, and we'll catch you on the next